All right, are there any questions? Any questions at all from last time? So I hope that you guys are reading, right? The material that has been given to you. I gave the lecture notes also, right? That's the material that you will be tested on, right, in the exam. Okay, so keep that in mind. So we are going to move on to the next chapter, which is energy considerations in biochemical reactions, right? So as I said, cells have to perform thousands of reactions every second in order to stay alive, right? And many of these reactions are like reversible, right? They could go in one direction or they could go in the other, right? What determines whether the reaction will go in a particular direction or not? Okay, right. So that's what we are going to discuss in this chapter, and we are going to revisit the laws of thermodynamics, the first law, the second law, and so on. Okay, because living things obey all those laws. Okay, and despite that, they are kind of producing order, right, in a universe that is tending towards disorder. Right? And let's see how that biological order is created. So we examine the factors that determine whether a particular biochemical reaction is feasible or not, right? And then the question is, if it is feasible, what additional factors are needed to get the reaction started, right? I give you an example, like if you take a piece of paper, right, and you set fire to it, okay, it burns and it is reduced to ashes, right? So that's a feasible, it's not a biochemical, but it's a feasible chemical reaction, right? But does that reaction happen spontaneously if you don't apply the match? If you don't apply the lighted match to it, it doesn't, right? So it needs, I mean, it's feasible, but it's not going to happen by itself, right? So it needs something else, right? So even for biochemical reactions, that is reactions that are taking place inside living organisms, right? There are energy considerations that determine, first of all, whether the reaction can take place or not. And then the second thing is what is needed to get the reaction started. Now it is a fact that the universe is always tending to greater disorder, right? So that is which law of thermodynamics? Second law, the entropy of the universe is increasing, right? So disorder is being created. Now if you look at living organisms, all right, order is being created and maintained, right? Like let's say from a single cell, you have a complete human being, right, with fingers in the right places, the head in the right place, and so on. Okay, even if you see a plant growing from a seed, right, with the root, the shoot, the leaves, and all that, it looks like order is being created. Okay. On the other hand, like if you don't clean your home or your house, right, it doesn't take very long before everything is messed up. Okay, for the non-living world, it seems to be tending towards disorder, right? So, so you might wonder, all right, are living things violating the second law of thermodynamics? And the answer is no, right? They actually obey the basic laws of thermodynamics. And again, to recap, the first law of thermodynamics is that energy can neither be created nor destroyed, and it can only be converted from one form to another, right? And one time when I was teaching this class, I got a protest from somebody about conversion of mass to energy and so on. Okay, we are not going to delve into all those things. We want to get just the big picture, right? Now the second law of thermodynamics says that the entropy of the universe can only increase, right? That means the disorder is only increasing, right? And the entropy, of course, is a measure of disorder. Now, at the same time, you're seeing that living organisms are generating order. So when a cell creates and maintains order, the disorder in its environment increases, right? And the sum total of the disorder in the cell and in its environment, that is going to increase so that the second law of thermodynamics is not violated. Okay. Any questions so far? Now, just in order to stay alive, right, living cells have to perform a never-ending stream of reactions because the cell has to be powered all the time. Okay. Right. It has to produce energy. Right. If it's a plant cell, it has to absorb energy from the sunlight and then produce sugars and things like that. Right. So they are performing a never-ending stream of reactions. So despite the diminutive size of the cell, it is performing thousands of reactions each second. Right. So each cell is like a tiny chemical factory, and thousands of reactions are performed. Right. Now, many of these cellular reactions do not normally occur at biological temperatures. Like you could put the reactants together and wait for thousands of years, no reaction will take place. All right. Yet in living organisms, these reactions readily take place, right, within a short period of time. Right. So such reactions are made possible by using catalysts, which are called enzymes, and coupling reactions together. Okay. So there are two things here. First of all, if you use a catalyst, okay, that can speed up the reaction, right? without itself undergoing any change, right? It itself doesn't get altered, but it helps the reaction. So catalysts and in living organisms, usually the catalysts are proteins, right? And they are called enzymes, right? So you've heard that term, but what is an enzyme? An enzyme is basically a catalyst, right? So that's one mechanism. And the other mechanism is that if you have, some reactions which will not normally occur, right? You can couple them with other more favorable reactions, right? To make them occur so that the combination is energetically favorable. And I give you an example. Like if you have like a mass, right, and you want it to move up, right, against gravity, it will not move up by itself, right? The natural tendency for it is to fall down, okay? But if you want to pull the mass up, right, what you can do is you can take a string, right, couple this mass to another heavier one, right, with the string, and pass the string over a pulley, right? Then if you release it, the heavier one is going to fall down, this is going to move up, right? So by that kind of coupling, reactions that would not happen otherwise, right, you can make them happen, right? By coupling it with some other reaction that is more energetically favorable, right? And the cells do that all the time, okay? Any questions? Now if you look at cells, right, whether it's in plants or in animals, there are two main types of chemical reactions in cells, right? The first set of reactions are what are called catabolic reactions, right? Where foodstuffs are broken down into smaller molecules, generating useful energy, right? So we get most of our energy from glucose, right? What is the formula for glucose? Anybody? Not you, not you. You have some bio background. Yeah, go ahead. Okay, good. Yeah. So that's the formula for glucose, right? And so glucose is oxidized, right? So C6H12O6, right? Plus 6 molecules of oxygen will give you carbon dioxide, right? I mean, you can balance the equation, but it's basically sugar, right? Plus oxygen will give you carbon dioxide plus water vapor, okay? So that's why, I mean, we basically breathe in oxygen, right? And we exhale carbon dioxide and water vapor, okay? So that's an example of a catabolic reaction where foodstuffs are broken down into smaller molecules and releasing energy. Now, one thing which I will emphasize again in a future slide, but I want to bring it up again right now, right? See, if you burn a piece of paper, right? You set fire to a piece of paper, energy is released, okay, but all of it is in the form of heat, right? Heat and some light, okay? But you don't have any useful energy left. Once the paper is burnt up, all your energy is gone. Okay? It's been released as wasteful heat energy. In the cells, that does not happen. The cell is going to try to absorb as much of this energy as it can, right? Or absorb it in a useful form. Okay? And that is made possible by carrying out these reactions. Let's say the oxidation of glucose to carbon dioxide and water. It is carried out over many incremental steps. Because if it releases too much of energy in one step, that cannot be absorbed, right? But if it's a little bit released, you can store it in ATP or some other molecule, right? And the cells do it that way, okay? And there is a future slide that I will get to, all right? Which says that about 50% of the bond energy in glucose, right, is converted into useful energy. If you compare that with an automobile engine, even the best automobile engine in the world can convert only about 20%. Okay. So the living organisms are a lot more efficient, right? So that's an example of a catabolic reaction, that is the oxidation of glucose. Anabolic reactions are reactions where you use the energy that is harnessed by catabolism to produce the biomolecules that the cell needs. Okay, like the cell needs to build up tissues and things like that, right? Bone, tissue, and so on. If it's a human cell, or in the case of plants, it needs leaves and so on. Okay, those have to be built up, right? So one example of an anabolic reaction would be like photosynthesis, right? Because there you're basically taking carbon dioxide and water, right? And with the help of sunlight, right, you're producing sugar. Like not glucose, you're going to produce sucrose, right, and starch. Okay, so that's an example of an anabolic reaction, right? And the catabolic and anabolic reactions together, they constitute the metabolism of the cell, right? Metabolism, you hear this term all the time. Okay, what does that mean? It's catabolism and anabolism together, right? So the biosynthetic reactions, right? And also the reactions that are associated with the breakdown of foodstuffs, right? Any questions? All right, now we want to introduce the concept of free energy, right? So we know from the second law of thermodynamics that the entropy of the universe has got to increase, right? So the reactions will proceed in a direction where the entropy increases, right? So entropy is a measure of disorder, right? So instead of looking at entropy, instead of looking at the increase in entropy, we can look at a reduction in free energy, right? Free energy is the amount of energy that is available to do useful work, right? So a reaction is going to be energetically favorable if it proceeds with a decrease in free energy, because the natural tendency of the universe is for the entropy to increase, okay? Now if you look at cells, they are generating biological order, right? Which is not going to be an energetically favorable reaction, right? It's an energetically unfavorable reaction because it seems to go in the opposite direction from what the second law of thermodynamics says, right? So cells generate biological order by coupling the biological order generating reactions, which are inherently energetically unfavorable, right, to other heat generating reactions which are energetically favorable. And I'll give you a few examples of that in this lecture itself, right? Now by releasing heat to the environment, right, the cell ensures that, number one, the entropy of the cell and its surroundings increases, right? And biological order is created within the cell itself, right? Okay, any questions so far? Yeah, go ahead. No, the releasing heat is an energetically favorable reaction because the entropy is going to increase, right? But the biological order generation, like if you're trying to synthesize, let's say, polysaccharides from glucose, right? Because those are, you're putting things together, you're generating order, right? Those are energetically unfavorable. So what this is going to try to do is to couple that reaction, right, with some other reaction that is releasing heat to the environment and is energetically favorable, right? And if the free energy change associated with that latter reaction is more, then this reaction, which would normally not have occurred, right, will also occur. And I'll give you examples of that, yeah.
How it is creating biological order? The last line says creates biological order within the
No, yeah, biological order is great. See, if you look at a living organism, all right, biological order means, you know, like if you look at a living organism from a single cell, all right, you get a complete human being, all right, everything seems to fit in and, it's happening properly. So that is creation of biological order. Or let's say a seed grows into a plant, okay? That is creation of biological order, right? I mean, that's a completely functioning thing that is practically built up from nothing, right? You started out just with a seed, or in the case of a human being, a single cell, okay? So order is being created. Or anytime you take, let's say, a molecule of glucose, all right? You're doing a condensation reaction. That is an order generating reaction because you are kind of making it more systematic, right? Instead of messing up everything, right? So that is an order generating reaction.
It says releasing heat to the environment ensures that biological order is created within the cell itself.
No. See, biological order cannot be created while violating the second law of thermodynamics, right? So the biological order is going to be created within the cell itself, right? But at the same time, heat is going to be released to the environment, right? So that is an energetically favorable reaction. So that will make sure that the combination of of whatever the reaction is that is generating biological order and this heat release to the environment, right, is together an energetically favorable reaction. So the
Overall, the entropy will increase.
yeah. Overall, the entropy is going to increase. All right. Any other questions? All right. So now we will look at some common biochemical reactions in cells. All right. And the first example is photosynthesis. All right. It's a process by which plants trap energy directly from sunlight to produce sugars. Right. So it's going to be carbon dioxide, right, plus water plus sunlight is going to lead to the production of sugars such as glucose, sucrose, starch, and so on. Okay, that's photosynthesis. The opposite of that is cellular respiration, where the carbohydrates are oxidized. Carbohydrates and sugars are oxidized to produce useful energy. Okay, and as I said before, the cellular respiration takes place in many steps, right? If it just, if in one step you just do, glucose and oxygen is going to give you carbon dioxide plus water, you will release some heat energy, nothing is going to get stored, right? The cell has to store the energy. Now let's define oxidation and reduction. I'm sure that you've seen that in your high school chemistry class, right? Oxidation reactions involve the addition of oxygen or the removal of hydrogen, right? And reduction reactions involve the addition of hydrogen or the removal of oxygen. Okay, and you can think of this also in terms of electrons. Okay, because hydrogen is like an electron donor, right? Oxygen is an electron acceptor, right? So loss of electrons is oxidation, gain of electrons is reduction. Right? Again, this is also something you must have seen in your high school class because you have done your high school many, many years after me, right? And I recall seeing that like maybe, some, I don't know, 35, 40 years ago, So let's take a little bit closer look at photosynthesis. Again, this will be the overall picture, but photosynthesis is one of the topics that I will cover in the last third of this course, right? Because the focus of this course, remember, is on plant genomics, right? So I mean, it would not be enough to just, cover photosynthesis using that one reaction. I have to tell you more things about photosynthesis, because if you're trying to increase biomass accumulation in plants, okay, or you're trying to increase the yield of plants, right? Then you need to know something about photosynthesis. What else is involved? Some details, right? I will cover those in the last third of the course. But for this chapter right now, right, the overall reaction of photosynthesis in plants is you have carbon dioxide plus water, and then you need sunlight, right? And that leads to the formation of sugars, and it releases oxygen and heat energy, right? And if, for example, if N is 6, then you will get glucose, right? C6H12O6, right? Now, photosynthesis is actually a two-stage process, right? In the first stage, energy from sunlight is captured and it is transiently stored in chemical bond energy in specialized small molecules, right? And what is one possible molecule in which bond energy could be stored? Again, not you, okay, I mean, what is adenosine triphosphate, yeah, ATP. Okay, that's one, and again, towards the end of this chapter, we will talk about adenosine triphosphate. So it can either be stored as high energy bonds in adenosine triphosphate, or you can store it in the form of high energy electrons, right? I will talk about that. There's a different set of carrier molecules where the energy is not stored as a phosphoanhydride bond, but as very high energy electrons. Because if you have high energy electrons, okay, they can supply reducing power. Remember I said gain of electrons is reduction, right? So if you have real high energy electrons, those will have that molecule will have a very, that particular carrier molecule will have a very strong tendency to donate those high energy electrons, okay? So it provides useful reducing power, right? So in photosynthesis, the first step is energy from sunlight is captured and it's transiently stored as chemical bond energy in specialized small molecules, right? And I will talk about these molecules as we go, uh, as we make a little bit more progress into this chapter. And of course, there's a lot more detail in the last third of this course, right? In stage 2, these energy carriers, like you have these molecules with high energy like ATP, and then you have some other high electron carriers. These guys from the first stage are then used to drive a process in which sugars are manufactured from carbon dioxide and water. Okay. And the sugars that are produced are used as, number one, a source of chemical bond energy, right? And also as a source of materials to make the many other organic molecules that the cell needs, right? For example, like if you look at wood, right? Wood is made of cellulose, right? And that is actually basically from the sugars, right? It's a polysaccharide, right? That is produced from the sugars, right? So, the cell, it produces a lot of different materials for which the starting point is the sugar. Okay, so both it gets the raw materials that it needs, and it can also be used as a source of energy. All right, any questions? And yeah, go ahead.
Is sunlight a kind of catalyst or
No, sunlight is a source of energy. Okay,
source of
so yeah, this is a biologically unfavorable reaction because again, this is going to generate order. Okay, and if you look at the free energy change, which I'll talk about, so this is unfavorable. So you need some external energy to get it going. Yeah, yeah. It is not sunlight. It cannot be a catalyst because the catalyst is some substance that helps the reaction happen, but will not get changed. Okay, sunlight is a source of energy, and we will talk about this. We will delve deeper into this, much deeper than I have gone into in earlier semesters because the focus this time is on plant biology. I mean, animal biology also will be covered, but this time in fact, I'm reading right now about photosynthesis, all those details. I've read it like once the entire book, and then I'm doing it the second time, and so I also have to work. It's not just you, right? Before I teach it in the last third of the course, okay, I would have had to read the entire book one more time and also made the notes. Okay, because I cannot come here and spend 2 hours, covering one chapter. Okay, I have to do it lot more efficiently. Okay, and that's good for you also, right? So yeah, we will talk about the details, but sunlight is the source of energy. You are going to need that energy to drive this reaction forward. Just like you cannot lift up, the weight will not spontaneously move up, right? If you have a weight here, you need to couple it to something else, right? So sunlight is providing that energy that is needed for making this reaction happen. And this reaction also, it doesn't happen in one step. There are a lot of different steps, okay? And catalyzed by different enzymes and all that. We'll talk about those things later, okay? Now for cellular respiration, that's just the opposite of photosynthesis, because here you're going to basically burn up the sugars or oxidize the sugars to get energy, right? So cells obtain energy by the oxidation of organic molecules. For carbohydrates, the general reaction is CH2O. That's the formula for a carbohydrate, right? Plus oxygen gives you carbon dioxide plus water plus energy. And again, keep in mind, as I said before, this reaction takes place in many steps, right? In fact, from the sugar you can get some, and this cellular respiration also I will cover in detail later, because it takes place in, if you're looking at glucose NS6, right? C6H12O6. So you want to oxidize glucose to carbon dioxide and water, right? So there are two major steps to it, right? The first step is where you split up a 6-carbon sugar, that is C6H12O6, into two 3-carbon sugars, right? That does not require oxygen, but it produces very little energy, and that's called glycolysis, all right? Which means basically lysis means to split up, right? Glycolysis means splitting up of sugar. So the first step is glycolysis, where no oxygen is needed, right? And then the second step is called the TCA cycle, or it's also called the Krebs cycle, which uses a lot of oxygen and it produces a lot of energy, right? Like if glycolysis produces 4 ATPs, this other one will produce 30 something ATPs, maybe 35 or 36. Okay, so in the presence of oxygen, the cells are, if there is no oxygen, then the cell will run glycolysis. If you have oxygen, then it will run both the glycolysis and the TCA cycle, right? So we will talk about these things again in the context of plant biology that we discussed in the last third of this course, right? Because even the process by which alcohol is produced and all that, that runs glycolysis along the way, right? From the sugar, how you get alcohol and so on. Okay, I'll talk about those. So this process over here, this overall reaction is referred to as cellular respiration, because oxygen is used up and carbon dioxide and water vapor are released. Okay, so it's not just that we as human beings, we breathe in oxygen and breathe out carbon dioxide and water vapor. Even at the cellular level, the respiration takes this form, right? And a great deal of energy is released, okay, when this happens, when you're oxidizing sugars. And as I've been emphasizing before, the cell would be unable to store this much energy if this process occurred in a single step, and therefore the reaction is carried out in several incremental steps, right? And each step results in the release of a small amount of energy that is stored either in the form of ATP, right, adenosine triphosphate, right, or even in the form of high energy electrons, right? And then the cell can store this energy in specialized energy carriers. And as I already mentioned towards the beginning of this lecture, about 50% of the bond energy is stored in a retrievable form by the cell, right? And it's a highly efficient process, much more efficient than any engine that has been designed by man. Okay. All right, any questions? Now let's again revisit oxidation and reduction. As I said before, oxidation is the addition of oxygen, removal of hydrogen, right? Reduction is the addition of hydrogen, removal of oxygen. All right, so if you're looking at this molecule of sugar getting converted to carbon dioxide and water, right? You can see that the sugar molecule is oxidized while the oxygen molecule is reduced, right? Because hydrogen got added onto the oxygen molecule to produce water, right? And whereas the sugar molecule, it got oxygen, more oxygen, and hydrogen got removed, okay, so it has been oxidized, all right? And I also mentioned that oxidation and reduction can be stated in terms of the loss or gain of electrons. Oxidation involves loss of electrons, reduction involves gain of electrons, right? And if you're looking at a chemical reaction, the total number of electrons does not change. So if one of the reactants gains electrons, the other one will have to lose electrons. Okay, so one will get oxidized, the other will get reduced. So they occur simultaneously in the same reaction, and these reactions are called redox reactions, right? Reduction oxidation reactions. Now the term, see, since oxidation and reduction involve loss or gain of electrons, right? Even when there is just a partial shift of electrons, you can think of oxidation and reduction, right? Because remember, I said earlier, right, there are covalent bonds, okay, where no electrons are actually lost, right, but the atoms, right, the atoms of different elements, they attract electrons to different degrees, right? That's how you get the hydrogen bonding in water, right? Because hydrogen has got a lower affinity for electrons than oxygen, right? Same way hydrogen has a lower affinity for electrons than nitrogen, right? Okay, keep that in mind. Whatever we see, like I'm not putting in unimportant details in the course, right? Whatever I have covered, I'm going to need it later. All the I wouldn't say it's unimportant, but all the stuff that is not important, let's say from an engineer's point of view, I have filtered that out. Because if you write my notes out, it's like 200 pages long, but that book is like 600-700. Not the plant biology book, but the original book that I used for the earlier class, that's essential cell biology, that's like 600-700 pages, okay, of type material. And mine would be handwritten notes, like maybe about 200 pages, all right? So don't try to, pick and cherry pick items here for study. For the exam, you will study everything, all right? No, not because I'm forcing you to study or to get a good grade, but because this is already filtered. You really need to know more, right, to be able to do useful research, right? Not less, right? This is already filtered, already cut down. I'm not a biologist, so I don't know all that is there is to know, right? But we are working at the interface of engineering and biology, so you need to know at least this much, right, if not more. Okay. So such a shift, all right, occurs when a carbon atom is covalently bonded to an atom with a strong affinity for electrons, for example, oxygen, right? And the carbon atom gives up more than its equal share of electrons, and it forms a polar covalent bond, right? And it is said to have been oxidized, right? And you can look at this reaction, right, which is basically the step-by-step oxidation of the gas methane to carbon dioxide, right? And no matter how you look at it, you will see that it is oxidation, because if you go from methane to methanol, okay, what does that involve? That involves the addition of oxygen, so that's oxidation. Methanol to formaldehyde, right, what does that involve? Removal of hydrogen, again oxidation. Formaldehyde to formic acid, oxygen, again oxidation, right? And then carbon dioxide, yeah. So again it's oxidation, right? If you again think of in terms of the electrons, right? Electron movement, if you're looking at the carbon atom in the center, you've now, if you go from methane to methanol, right, you have added oxygen. Oxygen has got greater affinity for electrons. So the carbon atom is losing electrons, right? I mean, there's loss of electrons, right? Or at least a partial shift. So it's getting oxidized. And similarly, you can look at the other steps and you can conclude that this is an oxidation reaction, right? Step-by-step oxidation. Any questions? Okay, now we are going to talk about enzymes, okay? Enzymes are proteins that make reactions possible, right? That would never have occurred, even in 10,000 years, right? So as I said before, chemical reactions proceed only in a direction that leads to a loss of free energy, right? And we know that from the second law of thermodynamics, because the entropy has to increase, right? Such reactions are said to be energetically favorable, right? So reactions that will just occur by themselves, right? That means with an increase in entropy, right? Or a decrease in free energy, those are energetically favorable reactions. Even these reactions do not occur spontaneously. And the example I gave you is, setting a piece of paper on fire. It does not occur spontaneously. You have to apply a lighted match to it before the thing will catch fire, right? So the molecules require activation energy, that is a kick over an energy barrier to get the reaction going, right? So when you apply a flame to that piece of paper, that will raise the temperature, right, enough to get the reaction going, right? So one way to overcome the energy barrier is to supply heat energy, right? But in the case of living cells, you cannot do that, okay? Because to get the reaction going, you'll kill the cell, right? So living cells have to operate within a narrow temperature range. And such a mechanism, it's just not going to work, right? You cannot raise the temperature high enough to get the reaction going. So the necessary boost over the energy barrier, in the case of living organisms, is provided by a specialized class of proteins which are called enzymes, right? And enzymes, these are very effective catalysts, right? Often speeding up reactions by a factor of as much as 10 to the power of 14. right? They are among the most efficient catalysts known in the world. Okay. And each enzyme will bind tightly to one or two molecules which are supposed to react, right? That are called substrates. Again, this is terminology. I mean, you can say you have to memorize it, but you have to be able to, speak the terminology. You have to know the jargon, right? Then only you'll be able to collaborate with people in different disciplines, right? Whether it is in medicine or it is in agriculture, right? They are not going to learn all the, KCl, KVL, and all that stuff. They will probably not be able to do because, many of them they have divorced themselves from math and the quantitative sciences pretty early on in their careers, right? So in their careers, so pretty early on. So they are not going to be able to learn like calculus and all that stuff. But we can do it. Okay, but provided we put in the work to learn all this terminology and all that, because this is like second nature to them. To them it's like ABC, right? Right? So that's one of the important things that is needed for cross-disciplinary collaborations. You have to get out of your comfort zone, learn all these things. And, this is an opportunity for you. I'm not giving you any homework. There'll be some, but it's a little bit homework. But learn all these things, and you have an exam to take, so, that'll motivate you to learn. Now, each enzyme binds tightly to one or two molecules that are called substrates. So this terminology is important. What is an enzyme? What is a substrate? Right? So this greatly reduces the activation energy of a particular chemical reaction. And once the substrates have reacted, then the enzyme is going to dissociate from the products, and it is free to bind additional substrate molecules. Right? So this is the same old story about the catalyst. It's helping a chemical reaction without itself undergoing any change. Right? It can be used over and over again. Any questions? So here is the picture, right? So let's say the reactants, let's say they are denoted by X, right? And this is the energy level that you're seeing, right? Along the y-axis, and along the x-axis, this is the reaction pathway, right? So you want the reactants X to react together, right, to produce the products Y. Okay. Now you can see that the free energy level of Y, right, is lower than the free energy level of X, right? So the reaction is energetically it's feasible, right? It is favorable. However, it has to climb over that hill in the middle. Okay, it has to go over that energy barrier. So if somehow you can push the reactants over that energy barrier, which is shown by the peak of the red curve, if you don't have an enzyme, right, the peak of that red curve is the energy barrier that has to be crossed. In the presence of the enzyme, right, is the peak of that blue curve that has to be crossed. So the height of the energy barrier has been considerably lowered by the presence of the enzyme. Okay, and that's how the enzyme is going to help the reaction take place. Because once you have helped these reactants cross that energy barrier, then the reaction is going to proceed spontaneously, okay, because it is energetically favorable. Now, enzymes are highly selective, and each enzyme is only going to catalyze a particular reaction, right? And by using different enzymes, a cell can regulate a number of reactions in a somewhat decoupled way. A cell that way is pretty intelligent, right? Because, and I'll give you an example. Like, see, normally, let's say if you look at a bacterial cell, normally it's feeding on glucose, right? So it is producing the enzymes that are needed for metabolizing glucose, right? Now, if you remove glucose from the environment, right, and you just put lactose over there, right, then the cell, in order to survive, it needs to produce the enzymes that are needed for breaking down lactose into glucose, right, and then it should be able to process it. And cells actually do that, right? This was discovered in the 1960s. I'll talk more about it later when I covered the chapter on gene regulation, right, and control of gene expression. This was discovered in the 1960s, right, and the people that discovered it, they got a Nobel Prize for that, right? So by using different enzymes, a cell can regulate a number of reactions in a somewhat decoupled way. Now, how do enzymes find their substrates? Right? Again, substrate is the molecule that the enzyme, or molecule or molecules that the enzyme will work on, right, or process. So enzymes will find their substrates through rapid diffusion. So inside the cell, you have all different kind of molecules that are moving around, right? So when there is so they'll randomly collide with each other. When there is no match, nothing happens. But as I said before, for proteins, it is the three-dimensional shape that determines everything. The next chapter is devoted to proteins, and we will discuss all these things in detail, right? But it's the three-dimensional shape that determines the property of the protein. So one protein, so the enzyme is a protein, the substrate may be another protein, okay? So the substrate has got a very special shape, right, that'll fit into the enzyme. Okay, it's like a hand fitting inside a glove, right? Hand in a glove type of thing. So when they find their perfect partner, that's when the reaction takes place, right? So random collisions between the molecules in a cell result in almost immediate dissociation, no reactions, right? However, when a substrate encounters the appropriate enzyme, immediate association takes place. Several weak bonds are formed, linking the two of them together and facilitating the necessary reaction, right? And once the reaction happens, then the enzyme is going to dissociate from the products, and again, it's going to get recycled. All right, any questions so far? So next, we move on to looking at the feasibility of chemical reactions, right? Now, chemical reactions are energetically favorable if they are accompanied by a decrease in the free energy. That is delta G, right? If G is the free energy, delta G is the change in the free energy. If delta G is negative, right, then that reaction is energetically favorable, right? Many reactions inside cells, for example, the synthesis of complex carbohydrates from sugars, or the formation of proteins from amino acids, you know that right now, I've covered that, or the formation of nucleotides, or the formation of DNA and RNA from nucleotides, those are all energetically unfavorable. So that they will not take place. Even if you wait 1,000 years, those reactions will not take place, right? So such reactions will occur only if they are coupled to an energetically favorable reaction such that the combined free energy change is negative, right? Think about that heavier weight being used to lift up that lighter weight, right, by using a pulley, okay? So adenosine triphosphate, or ATP, which is a molecule that I introduced last time, but in a very, very rough, sketchy way, right? Is a molecule that participates in many coupled reactions. And as I said last time, ATP is capable of storing a large amount of energy in its phosphoanhydride bonds, right? It's like the currency of the cell. ATP to ADP, right, or even sometimes ATP to AMP, right, releases large amount of energy, right? So this is like, charging a battery and discharging it when you need the energy. So again, if you didn't catch it last time, here is the structure of ATP. You're going to have the base. The DNA base is going to be adenine, okay, at the top, right, which has got a 9-member ring, right? I introduced all these last time, like purines, pyrimidine, and so on, right? And you have to keep on repeatedly looking at that stuff, right? Because that's how you will learn. There are no shortcuts here, all right? So that adenine again is connected to the 5-carbon sugar, right, which is ribose, right? And again, if you remember from last time, we label the carbon atoms as what? On the ribose we label them as 1 prime, 2 prime, 3 prime. At the 5 prime end, there is a phosphate that is attached. Okay, that's what gave you a nucleotide. In the case of, so that would give you adenosine monophosphate, right? Adenine plus ribose plus 1 phosphate is adenosine monophosphate. You add a second phosphate to it, you will get adenosine diphosphate. Add a third phosphate to it, you get adenosine triphosphate, or ATP, and it has got 2 high energy phosphoanhydride bonds. Okay. So ATP is obtained by adding 2 inorganic phosphate ions in cascade to adenosine monophosphate, or AMP, and one or both of these phosphoanhydride bonds can be readily hydrolyzed. You add a water molecule, you can break that bond, right, and release a large amount of energy, right? So it can be hydrolyzed to yield either adenosine diphosphate if you just break only one phosphoanhydride bond, or adenosine monophosphate, and both these reactions are energetically favorable, right? So these will happen spontaneously, right? ATP will have a tendency to, to basically go to ADP or AMP and release that energy, right? But if you don't couple it with some other useful reaction, that energy is going to be released in the form of heat. So now we get to an example of a coupled reaction, right? And that goes back to Rajan's question about generating biological order, right? You'll see how using ATP you can actually generate biological order. So if you look at the reaction that is used to form sucrose from glucose and fructose, right? I talked about these last time, right? Glucose, and then I mentioned also fructose, right? So if you basically link these two monosaccharides together, you get this disaccharide that is called sucrose, right? However, this is an energetically unfavorable reaction, right? The standard free energy change of this reaction is 5.5 kilocalories per mole, right? And again, delta G is the free energy change, delta G superscript O is the standard free energy change. See, the free energy change depends on the concentration of the reactants and the products, right? So the standard free energy change is basically when the reactants and the products, they have the same concentration, right? So the concentration term drops out from that equation. I'll show that to you later, okay? When the concentration term drops out, that's when you have the free energy that you get when it doesn't really depend on the concentration. That's the standard free energy change, right? So both reactants and products have the same concentration. Now, since this number is positive, this reaction is not going to occur by itself, right? But on the other hand, the hydrolysis of ATP is an energetically favorable reaction which has got a standard free energy change which is large and negative. It's minus 7.3 kilocalories per mole, right? So if you can find a way of coupling these two reactions together, right, then you will be able to form sucrose from glucose and fructose, right? Although this reaction is energetically unfavorable. And that's what happens in plants, right? Because plants produce sucrose, right, and starch. So the two reactions above can be coupled together as follows. First, the ATP is readily hydrolyzed to ADP, right? So an inorganic phosphate ion is released, but that actually goes into location 1 on the glucose molecule. Remember, glucose is a 6 carbon sugar. So you have C1, C2, C3, C4, C5, C6, right? So ATP is easily hydrolyzed because that's an energetically favorable reaction that produces what is called glucose 1-phosphate. That means glucose with a phosphate attached at the 1 location, right? The carbon on the 1 location. So the first reaction that you get is glucose plus ATP is glucose 1-phosphate plus ADP, right? Now that glucose 1-phosphate, the bonding of the phosphate with the glucose, that's a high energy bond, okay? So the glucose 1-phosphate will then react with fructose, right? With fructose to produce sucrose, right? And then that inorganic phosphate is going to be released. So the free energy change for sequential reactions is additive. So if you have if you have the products of one reaction driving serving as the reactants for the next one, you can add the free energy change, right? So in that case, the standard free energy change for this overall reaction in these two steps is actually negative. It's minus 1.8 kilocalorie per mole, right? You just add those two numbers, right? Which was, what was the number? 5.5 minus 7.3, right? You got minus 1.8 kilocalorie per mole, right? So the net result is that this overall reaction is energetically favorable and it will take place, and sucrose can be made in a reaction driven by the hydrolysis of ATP. Okay. Any questions? All right, as I promised you earlier that I will talk about standard free energy change, okay? So the free energy actually depends on the concentration terms also, right? And when the effect of the concentration terms cancels out, then the free energy change that you have left, that is called the standard free energy change. So the free energy change for a reaction depends on the concentration of the reactants, right? So consider a simple reaction below, right, where you have basically one reactant, which is A, right, is getting transformed into the product B, right? The free energy for this reaction is given by that formula. Delta G is delta G superscript 0, the standard free energy change, plus 0.616 natural log of the concentration of B over the concentration of A, right? And here delta G superscript O is the standard free energy change. B with the square brackets is the concentration of B in moles per liter, and A is the concentration of A in moles per liter, right? And you can see from this equation that if B and A have the same concentration, all right, then the concentration term drops out because you have natural log of 1, which is 0, right? And the free energy change that you get is the standard free energy change. Okay. I think so, yeah. Yeah. It's a factor. I mean, maybe it's from RT and things like that, you know. I mean, you have to look at a book on chemistry to see what it is. But again, I mean, this stuff you have probably studied in inorganic chemistry, like the rate of a reaction, right? is proportional to the concentration of the reactants, right? So you have some constant multiplied, so the rate of the forward reaction will be some constant multiplied by the concentration of the reactants. Then for the reaction proceeding in the reverse direction, again it will be some constant multiplied by the concentration of the products. We will do that in the next chapter, a little bit of that, okay? But we're not going to delve that deep into it, just using the results, right? So, and then from there you can get the equilibrium constant and so on. Okay. So I will try to tie this to the free energy. Okay. The concept of equilibrium constant and all that, I'll try to tie it to the free energy to show you that if the equilibrium constant is large, right, then the reaction will go heavily in the forward direction, right, and so on from these equations. Now, if you are at equilibrium, right, that means that as much of the reaction is proceeding in the forward direction as in the reverse direction. So that means the free energy change for that reaction at equilibrium is going to be zero, right? Right? I mean, you start out with negative free energy, but as time goes on, let's say if the reaction is energetically favorable, A is getting converted to B, right? And then at some point in time, the free energy change for the conversion from A to B is zero. That's when you're going to attain equilibrium. So if you set delta G equal to zero, then you can solve for the standard free energy change. That is going to be delta G superscript 0 is minus 0.616 natural log of B over A. Equilibrium concentration of B over equilibrium concentration of A. Right? So at equilibrium, the concentration of the reactants and products does not change. Hence delta G is 0. And if A sub E and B sub E denote the equilibrium concentrations of A and B respectively, then delta G superscript 0 is minus 0.616 natural log of B, natural log of concentration of equilibrium concentration of B over the equilibrium concentration of A. Okay, and this quantity K, which is the ratio of the equilibrium concentration of B divided by the equilibrium concentration of A, is called the equilibrium constant of the reaction, right? So you can see, right, if the equilibrium concentration is large, right, if K, I'm sorry, the equilibrium constant is large, K, that tells you that the equilibrium concentration of B is much larger than the equilibrium concentration of A, right? Which means that the reaction is going to proceed in the direction, strongly in the direction from A to B, right? But this also ties into the notion of negative free energy, right? Because if B is much larger than A, right, the natural log over there is going to be a positive quantity, right? Right, and so free energy is going to be negative, right? So all these ideas, they actually gel together, right? The reaction will go in the forward direction if the free energy change is negative, right? The more negative it is, the higher you'll have the equilibrium concentration of the product. All right, any questions? Now, cells can speed up reactions in at least two ways, right? What you can do, one way is compartmentalization, right? So if you have a reaction that is going from A to B, right? So A is the original reactant, B is the product, right? So as the reaction proceeds, more and more of B builds up, okay? And that has a tendency to drive the reaction in the opposite direction, to slow the reaction down, overall reaction down. So what the cell could do is, as the product B is being produced, it can remove that, right? So it will keep the reaction going in the forward direction. So cells do that. So they rapidly remove the products into a separate compartment, right, to keep the reaction going in the forward direction. Then the other thing that the cell can do is to use up the products of one reaction as the reactants of an immediately following reaction, right? So that way also you're decreasing the concentration of the product, right? Because this product is getting used up as soon as it is being produced, right? So the reaction keeps moving in the forward direction. Moreover, in this second case, the cells can cause the energetically unfavorable transition, let's say from X to Y, to occur if an enzyme catalyzing the X to Y reaction is supplemented by a second enzyme that catalyzes the energetically favorable reaction Y to Z. Right? X goes to Y, right? That might be energetically unfavorable. You used an enzyme, right? And then Y to Z, you immediately convert Y to Z, right? So if the overall free energy change for the reaction from X to Y and then to Z, right, is negative, then the reaction X to Y will proceed rapidly, although by itself it would have been thermodynamically impossible, right? Cells do that all the time because they have to, if the cell needs something, it has to produce it, right? Even if it is even if the production is an energetically unfavorable process, right? So the living organism has to figure out a way to make it, and they will do that by coupling or by using up the reactants to drive a more energetically favorable reaction. Any questions? So now I want to talk about activated carrier molecules and their role in biosynthesis. Right? Activated carrier molecules, basically they can transport energy, right, from one location to the other, from the site of generation to the site of utilization. Right? So the energy that is released by the oxidation of food molecules is used in the construction of other organic molecules. And the energy is temporarily stored as chemical bond energy in what are called activated carrier molecules or coenzymes. Right? It's just like generating power, okay? You have to either use power or you have to store it. Right? You have to store it in a battery or some kind of cell. Right? So these molecules, these activated carrier molecules, they transfer energy from one site to another by rapid diffusion. So it's generated in one location and the energy is needed somewhere else. These molecules move around and then the energy can be extracted at the appropriate location. So the coenzymes store their energy either in high energy bonds, right? Example, phosphoanhydride bonds that we have seen in ATP, right? And even other molecules like GTP is another molecule, right? It's not, extensive. It's not used that much, but even GTP also will have high energy phosphoanhydride bonds, right? Guanosine triphosphate, right? Or it'll store the energy as high energy electrons in another class of molecules, right? And that class of molecules also is very important, and I will talk about them right now, right? So first let's finish up the discussion about ATP. So ATP, or adenosine triphosphate, is the most widely used activated carrier molecule in cells, and the hydrolysis of ATP releases energy for doing cellular work and also for biosynthesis. Like if you're trying to produce sucrose, right, from glucose and fructose, right, you need energy. And the reverse reaction, that is forming ATP, is driven by energy from sunlight, right? When you're doing photosynthesis, ATP is formed, right? Or by the oxidation of glucose. When you oxidize glucose, right, again the energy that is released, that has to be stored in some energy carrier. So it's going to be released incrementally, but it's going to be stored in ATP, right? So this ATP-ADP cycle, right, that is the mechanism by which our cells are powered, right? So in the low energy state in a cell, you'll have lots of ADP. You want to increase the energy level, convert the ADP into ATP, right? This is called the process of phosphorylation, where you add a phosphate group. So ADP is phosphorylated to ATP to build up energy, right? When you need energy for something, ATP is basically hydrolyzed, right? That means broken up with the addition of a water molecule to produce ATP, and the energy that is released, it does cellular work. Even that's the mechanism by how our muscles and all that move, okay? Because you need energy to move, right? I mean, so there has to be ATP molecules have to be broken down in your muscle cells, all right, and tissues to supply the energy that is necessary for you and I to move. Now another class of activated carrier molecules are what are called NADH and NADPH, all right? So I'm going to show you the picture first, because otherwise just going through this text over here is not going to make any sense. I'm going to show you the picture. The picture is a little bit intimidating, but don't get scared. I'll explain to you what is important. Okay, all of this picture is not that important, right? There are details here, but what the key issues are, right? So this is a molecule, right, that is called, on the left hand side it is called NADP plus, right, and that is nicotine amide dinucleotide, no sorry, nicotine amide adenine dinucleotide phosphate, right, NADP, okay. Don't worry if you cannot remember all of this after today's lecture, but try to pick it up, in the course of this class, right. So it's nicotine amide adenine dinucleotide phosphate, okay, that's on the left, all right. And on the right, that is called nicotine amide adenine dinucleotide phosphate reduced, because it has the H on it, all right. So what's the difference? What's the difference between the left and the right? Well, if you take that molecule on the left, all right, and you add to it a proton and two high energy electrons. A proton is a hydrogen ion, right? A hydrogen atom, if it has lost its electron, it's a proton. So you add a proton and two high energy electrons, okay? So that is denoted by what is called the hydride ion, which is H minus, all right? If you add it to the stuff on the left, okay, you get what you see on the right, right? Because so now that plus sign on that N is also gone, and you actually have on the right at the top, you see those two, the cross over there, right, between those two H's. That has got those two high energy electrons. So it can supply reducing power, right? And so on the left you have so this is called nicotine amide adenine dinucleotide phosphate, all right? So at the top, right, see that ring that you see, that's the nicotine amide ring, right? The ring at the top that you see in each of these pictures. Dinucleotide, because you have two of them, right? I mean, you have adenine here, you have ribose, right? This is just like the sugar phosphate backbone that I covered last time when you're linking nucleotides together, right? Remember the 5' end of the phosphate at the 5' end is going to link up to the 3' end of the next sugar, right? That's how DNA is produced, you know. But here I think both of them have the 5' end only, the linkages, all right? But so nicotine amide, adenine is there, so it's adenine, dinucleotide, all right? And then you have this phosphate at the bottom, okay? So this is denoted by NADP plus, right? Because there's a plus sign on that nitrogen, you see, right? If you add to that a proton and two high energy electrons, you get the stuff on the right, which is called again NADPH, but it's nicotine amide adenine dinucleotide phosphate reduced, right? Okay, and that can supply reducing power because that has absorbed high energy electrons. It can give those up when needed for some reactions. Okay, and typically this molecule is used a lot in biosynthetic reactions like photosynthesis, NADPH. So in photosynthesis, the two carriers that will be important are ATP, right, and NADPH, because NADPH can provide the high energy electrons that are needed, right, for carrying out the reactions of photosynthesis, right. So keep that in mind. Now, if you have exactly the same molecule that you see on the left and on the right, but with the phosphate group taken out, right? This thing at the bottom is not there. The phosphate group is not there, right? Then you call that NAD plus. So NADP plus without the phosphate is NAD plus, right? So again, it's nicotine amide adenine dinucleotide, right? No phosphate, right? And on the right, again, if the phosphate is not there, it's just going to be NADH, right? Nicotine amide adenine dinucleotide, right? And it's reduced. H means reduced. Again, the stuff on the right can supply high energy electrons, right? But there are two sets of these carrier molecules, right? One set has a phosphate, the other doesn't. The one which has a phosphate, that's the one that I'm showing in this diagram, that serves to supply high energy electrons for photosynthesis and for biosynthetic reactions, right? Biosynthetic reactions. Whereas the other one supplies reducing power, right, for catabolic reactions, like where you break up glucose into to finally get carbon dioxide and water, right? So these are two carrier molecules. Now, don't ask me why is it so. That's the way it is, okay? And why did these nucleotides appear here? I don't know. But the good news is that all of this other stuff is unimportant, right? The important thing to note is the difference between the two, between the one that has a phosphate and the one that doesn't have a phosphate. That's number one. And the fact that you can go from the oxidized form, that is like NADP plus here, to the reduced form by the addition of a hydrogen ion, right, and two high energy electrons, right? So if you have high energy electrons, all right, you can start from the oxidized form, right, and supply those high energy electrons. You get the stuff on the right. That now has those high energy electrons. It can move around, and where the high energy electrons are needed, it can supply it. Okay, that's the important thing to know, right? Again, I mean, the whole figure might be confusing and all that, but you know, the rest of the details are not important. But these are the very, very important carrier molecules that take part in photosynthesis and in respiration. The one with the phosphate takes part in photosynthesis and other biosynthetic reactions. The one without the phosphate, that basically takes part in catabolic reactions like respiration and so on. Okay, so now this is a text that is describing that stuff. So picture is worth 1,000 words. So you know, we've already covered more than 1,000 words. So here in a few words we'll explain, and I think you will see that it makes sense. All right. So NADH and NADPH, right? Again, NADH is the one that does not have phosphate, right? So nicotine amide adenine dinucleotide, right? H is reduced, right? And NADPH is nicotine amide adenine dinucleotide phosphate reduced, are important electron carriers, right? The stuff on the right, I said it carries high energy electrons, right? They can supply reducing power needed for certain reactions. Then NADP plus, that's what we had on the left, is the oxidized form of NADPH. That means it doesn't have the high energy electrons, right? And NADP plus and both NAD plus also, they contain a nicotine amide ring, I showed you that, and it's called nicotine amide adenine dinucleotide phosphate, right? And by adding a proton, hydrogen ion and two high energy electrons to NADP plus, one obtains NADPH. That's what I showed you in that picture, right? And NADPH provides strong reducing power. Again, that's something that I've been emphasizing. Those high energy electrons will provide strong reducing power. And NADH and NAD plus differ from NADPH and NADP plus respectively in that the bottom phosphate group is missing in the former. Right. So I've explained everything. Using that picture, whatever is written here has already been explained. Okay, any questions? And these things, don't forget about them. Right. The first time I taught this course, I didn't emphasize all this. But remember, this time we are going to tie things together with plant biology. Right. And I will cover respiration in detail. Right. So there these carriers are going to be important. If you're looking at glycolysis, the TCA cycle and so on, and even in the different stages of photosynthesis, these carriers are going to be important. The one with the phosphate will be used for photosynthesis. For respiration, it is going to be the one without the phosphate. So you will see them again. And ATPs also, you will see them. So as I mentioned, NADH has a role as an intermediate in the catabolic system of reactions that generate ATP, whereas NADPH operates with enzymes that catalyze anabolic reactions, supplying the high energy electrons that are needed to synthesize energy-rich biological molecules. Right? An example would be photosynthesis. Right? So there are two different sets of activated carrier molecules for two different pathways. So thus the cell has got some amount of independent control over these pathways. Okay. It's not like if it tries to run respiration, automatically photosynthesis will turn on or something like that. There's the independent control, some kind of decoupling. All right. So we talked about the activated carrier molecule ATP. Then we talked about NADPH, right? NADH, right? Now there's another important activated carrier molecule that is called acetyl coenzyme A, right? That's also very important, right? In respiration and all that, acetyl coenzyme A occurs over and over again. In fact, see, the acetyl coenzyme A is the molecule, okay, where the pathway for oxidizing glucose, right, meets the pathway for oxidizing fatty acids, right? Our cells can get energy either by oxidizing glucose, all right, or it can get from proteins too, or from fatty acids, right? Remember we said earlier that fatty acids are much more compact storage of energy, right? They produce a lot more energy, right? However, the pathway through which you oxidize fatty acid is different from the pathway through which you oxidize glucose, right? So glucose initially, as I said, if you're trying to initially when you're trying to break down glucose, you do it without oxygen, right? The first step is called glycolysis, which splits up the 6 carbon sugar into 2 3 carbon sugars called pyruvate, right? So from the initial processing of glucose, you get pyruvate. Then this pyruvate is actually converted into acetyl coenzyme A, right? And that's the thing that is processed in what is called the TCA cycle, right? Or the Krebs cycle. Now, if you're trying to metabolize fats, okay, you're trying to break down fatty acids, by oxidation of fatty, you get acetyl coenzyme A. So the acetyl coenzyme A is the common point, okay, where both these pathways actually meet. Okay. After that, you don't know whether this thing came from glucose or it came from fatty acid, right? It's going to produce more energy, but acetyl coenzyme. So it's a very, very important molecule, right? When you're looking at, the fatty acid metabolism, right? And even the glucose metabolism of cells. Okay, and this is the common molecule where everything meets, right? So acetyl coenzyme A is another important activated carrier molecule, and the bond between the acid, and again I think the best way to explain is through a picture, right? So this is the schematic structure of acetyl coenzyme A. On the left, you can see that you have CH3CO, right? That is the acetyl group, right? C double bonded with O, that's the acetyl group. So that has been linked, all right, through a high energy bond, right, involving sulfur, right? So that's like a thioester bond, right? Anything involving sulfur, they call it thio, right? So it's a thioester bond. It is linked to the coenzyme A molecule on the right, right? So this, on the right of this high energy bond, you have the coenzyme A. Again, and there is a lot of detail to this structure, which is not important for us, right? This is the coenzyme A. When that links up with the acetyl group through this sulfur bond, right? This thioester bond, you get this molecule that is called acetyl coenzyme A. Now, what is so special about this? Well, this bond involving that sulfur and that carbon is a high energy bond, right? That means that acetyl coenzyme A, okay, if that acetyl group is needed for some reaction, right, the acetyl coenzyme A can move around readily in the cell, all right? Then if it gets to a location where the acetyl group is needed, this bond can be readily broken because this is a high energy bond. In an energetically favorable way, this acetyl molecule can be gotten rid of, all right, and it can supply the needed acetyl molecule for some other reaction, right? And this plays an important role in the TCA cycle, right? The Krebs cycle, right? That we will also talk about. Yeah, go ahead.
So by high energy bond, we are meaning it's a weak bond, right?
No, high energy bond means
Which
no. It's that means if you break this bond, right? This bond has a strong tendency to be. I wouldn't say it's a weak bond, but this tendency has got this bond has a tendency to be broken, right? And it will release energy. Okay, it's energy. The breaking of this bond is energetically favorable, right? Just like ATP, right? Because so that if that molecule is needed, this bond can be broken just like this, and that molecule can be provided for that reaction. Because in some of the steps in the metabolism of sugars and fatty acids, you need an acetyl group, right? In the TCA cycle, you need an acetyl group, and this one supplies that.
This means a source of energy.
Energy also, yes, yeah. It can be used, yeah, it has high energy over there, so yeah, it can be as a source of energy too, right? Because see, it's not just, look, when this bond is broken, all right, this acetyl group needs to get off this, okay, and get latched onto some other molecule, right? That is the biologically, order generating reaction. So that reaction would not happen by itself. If you just had this acetyl group sitting around and, going somewhere, it needs to latch onto something that will not happen, okay, because there's some energy barrier that has to be crossed. But by having this thing linked in that high energy bond, right, the breakdown of this, right, has got a high negative free energy, right? So this will drive that other reaction where this acetyl molecule can be latched onto the other molecule that it needs to latch onto. Yeah. Just like I showed you the formation of sucrose from fructose and glucose, right? It's the same kind of thing.
We have same meaning of NADPH. It's the same to the NADPH.
Yeah, yeah, no, those are not high energy. Those are high energy electrons, right?
Very real source of energy.
No, there it will be a source of, yeah, the electron has got high energy, but as I said, gain of electrons is reduction. So if in a particular equation, in particular reaction, you need reduction to take place, right? So high energy electrons have to be supplied for that reaction to take place. And this one, so the energy here is stored in a high energy bond, right? Whereas in the case of NADPH and NADH, the energy is stored as very high energy electrons, right? So some reactions might just need this energy or something to be transferred. Some other reactions may need high energy electrons. They might need a lot of reducing power, right? And if you go look into the detailed reactions of photosynthesis and respiration, right? At different stages. Remember, I said that the reaction is going to be carried out incrementally in many different steps so that the energy is absorbed, right? So some steps over there will, make use of ATP. Some steps will make use of these high energy electron carriers. Yeah. Okay, any other questions? So the conclusion of this chapter can be summarized as follows. So all the activated carrier molecules discussed, they contain a nucleotide. Right? Now I don't know why that is the case, but that, because see again the nucleotide has got the function of coding like DNA, RNA, I can understand, okay, but these activated carrier molecules, they have, it's like a that A sticking in there in ATP, right? Then when we looked at NADPH, right? Again, you had a nicotine amide ring and you had an adenine, right? For some unknown reason, that stuff is there. The nucleotides are there. And in fact, among these, ATP is the only one which is involved in actual nucleic acid synthesis, because you will basically link, see, even the way the nucleic acids like DNA and RNA are formed is that ATP has got those two high energy bonds, all right? You'll break those, you'll get AMP, all right? And then you link up, you'll create that phosphodiester bond that is linking one nucleotide to the next one. Okay, so like we don't know, for some reason, okay, I mean during evolution these nucleotides remained, right, in these carrier molecules, right, but they don't seem to be playing any particular function, but they are part of those activated carrier molecules. And the important role that is played by activated carrier molecules in biology is that the condensation reactions, all those energetically unfavorable reactions, are driven by these activated carrier molecules. So the condensation reactions involved in the synthesis of polysaccharides, okay? So starting from the simple sugar like monosaccharides, you want to get disaccharides, trisaccharides, polysaccharides, so produce starch, then produce cellulose and so on. Those reactions are energetically unfavorable. So they have to be powered by the energy driven from ATP hydrolysis. Same thing about the formation of proteins. Okay, remember last time I covered those amino acids. Amino group links up with the carboxyl group. The standard free energy change of that is positive, right? So you need the energy from ATP hydrolysis to drive that. Same thing about formation of nucleic acids from those modular units, right? Okay, any questions? Otherwise, let's get, yeah, we have a few minutes left, so let me get started with the next chapter. If there are no questions, all right, I will get started with proteins, all right. So proteins are the most versatile functional molecules that are found in a cell, right? So almost anything that, I mean, not everything, but almost anything that you can think of, the protein is responsible for that function. So they are responsible for carrying out nearly all cellular functions, and they constitute most of the dry mass of the cell. Let's look at some examples of proteins. Proteins that are called enzymes, they promote many chemical reactions. We already discussed that. Okay, proteins serve as catalysts. In that role, they are called enzymes. All right. Then proteins serve as signaling molecules from one cell to the other. If a particular cell is supposed to divide, it will get some protein from another cell directing it to divide, and so on. And those of you that attended my seminar last Friday, you saw me talk about that. Right? Proteins serve as moving parts of tiny molecular machines, right? Proteins can also walk like you and me, right? Then if you look at fish in the Arctic Ocean, they use proteins as antifreeze, right? So antibodies, right? These are proteins that protect us from infection, right? That build up in your immune system. Those are proteins. Toxins are proteins, right? If you look at snake venom, that is also proteins, right? Very efficient at killing people and basically stopping the cellular processes. Hormones, these are also proteins, right? Insulin is a protein, right? And so on. And the list goes on and on, right? Again, for proteins, shape is everything, right? The function of a protein is determined by its shape. It has to have exactly the right shape for its particular function. So proteins adopt a large number of different three-dimensional shapes. Thus, they can perform versatile functions. And the shape of a protein is specified by its amino acid sequence. Right? So we saw during the last lecture that a protein is made up of amino acids that are linked together. Right? So you have what is called a polypeptide backbone. Right? The alpha carbon, the hydrogen and all that stuff. Polypeptide backbone, and then for each amino acid there is a side chain. Right? So depending on the side chains that you have in a particular protein, right? That particular protein is going to fold up. It will have a specific shape, very much tailored towards its function, right? So a protein molecule is made up from a long chain of amino acids, and each amino acid in the protein molecule is linked to its neighbor by what is called a peptide bond. We have already seen that in the last chapter, right? Or in the last class. And the repeated sequence of atoms along the chain is referred to as the polypeptide backbone, right? Because the side chains are different, but other than that, the amino acid, if you forget about the side chains, all the amino acids look the same, right? And the side chains are unique to each protein, and they are crucial in determining its distinctive properties, because some of the side chains might be hydrophobic, they hate water. Some others might be hydrophilic, some might be charged, some might be might be uh, polar, some might be non-polar, and so on. Okay. Now proteins will fold up into different shapes because of different sets of weak non-covalent bonds, right? Such as hydrogen bonds, right? Right? I mean, because it might have a polar side chain where there is some uneven charge distribution, right? And across thousands of these molecules, right? That's going to make a difference. So hydrogen bonds. There could be ionic bonds, right? There could be charged side chains, right? Then there could be van der Waals attractions, okay? Which is basically, if you bring the atoms too close together, they're repelling, right? If you take them too far away, they're attracting. So it has to be at the right distance, right? Then hydrophobic forces, okay, because the protein might have hydrophobic side chain that hates water, so it'll try to stay away from water. So it'll fold up into a particular conformation. So consider the hypothetical protein in the following figure, which is made up of 4 amino acids, 2 polar and 2 non-polar, and you can see what conformation it will have in an aqueous environment. This is a hypothetical protein made up of 4 amino acids, like 2 of them are polar, right? That means they love water, 2 of them hate water. So chances are that if you had such a simple protein, you put it in an aqueous environment, right? Then the portion that hates water is going to stay away, right? While the portion that loves water is going to be facing the water, okay? And there are so many different forces that will finally determine the three-dimensional conformation of the protein. Now, in general, a given protein will fold into a conformation of lowest energy. Right, that's the most stable conformation. And a protein can be unfolded or denatured by treatment with certain solvents. Like if you treat it with urea, you can, even if it's folded in the three-dimensional shape, it'll basically fold out, right? Now when the solvent, however, the shape is very, very inherent to the amino acid sequence of that protein, because when you take the solvent away, the protein will again spontaneously fold into its original conformation. And then you have other proteins that are called chaperone proteins that will assist protein folding inside cells by speeding up the folding process. Right? So chaperone proteins do not alter the final three-dimensional shape of the protein. That is determined by its amino acid sequence. Right? But they can speed up the actual folding into the three-dimensional conformation. And as I mentioned last time, this determining the three-dimensional shape of a protein based on its amino acid sequence is a hot topic. Right? And if we figure out how to do that, right, we will make tremendous advances in improving the efficacy of drugs and things like that. Okay, because we will know exactly what set of amino acids to put together to make the final drug have exactly the right shape to bind the molecule that we are targeting. That's still an open problem. I mean, I don't recommend that you do it for your PhD because you may be doing a PhD for a long, long time, unless you find some subset of that problem to work on. Right? This is open, right? Now, proteins range in size from about 30 amino acids to more than 10,000 amino acids long, right? And majority of the proteins are between 50 and 2,000 amino acids. And the conformation of a given protein is very complex because so many different factors come into play, right? However, the good news is that there are few common structural motifs that underlie the various conformation, and this will facilitate a general study of protein structure. So instead of like if a protein is 10,000 units long, you don't have to study all the 10,000 units, all the possibilities in a different way, because there are probably some modules, okay, that I will talk about next time, right? Some modules, so instead of looking at the entire protein, the final three-dimensional shape is determined by a number of modules that are fitting together. Okay, parts of it will fold in one way, parts of it another way, and then everything comes together. Right? Okay, so let's stop here for today. Right, we're running out of time. So next time I will talk about those different modules that you have.